Hello, everybody. How are you? Ready for today's lecture? So today is lecture 25. Can you hear me and see me? Yes? Someone, please say something.
Yes.
Okay,
Yes.
great. I'm sure you all missed me so much. So let's see. So we have 28 lectures, right? And one of them is a movie. It's called The Case of the Frozen Addicts. And you can see it at home. It's about 55 minutes. It's very interesting. It's highly relevant to what we are covering now. It's sort of a detective story about discovering why these patients have these symptoms. So I don't want to say anything, but it's so I don't break the interest. So I don't want to spoil the story. So there will be two questions on the exam on this movie. So I highly recommend that you will see it. Get a bucket of popcorn, relax, and watch the movie. It's a good one. Okay, so what are we going to cover today? Today we're going to concentrate on the functions of the cerebrum, and then we're going to go through the thalamus and hypothalamus and so on, and cerebellum. It's all about brain function. I think it's going to be interesting. Can you have an exam review? Yes, absolutely. We have to decide what is the best time. So maybe on Thursday we'll discuss this. Probably going to be, so the exam is on the 16th, sorry, which is a Tuesday. So we probably can have a review on Thursday, the 11th. But let's, I'll confirm this in next class. Okay. So the functions of the cerebral cortex. So here you see the brain. Now you know how it's covered with the meninges, and you have the cerebrospinal fluid flowing around it and inside in those ventricles and the subarachnoid space. Right, we're not going to cover questions that are on the exam, but similar ones. Okay, so now, what really does the brain do? The cerebral cortex. So now think about a room in your house, and it has different light fixtures. Let's say it has 5 different light fixtures, and you have a switch, several switches, and depending on which switch you turn on, there's going to be light fixture A. So switch number 1, the electrical current is going to end up in light fixture A. So when you turn on that switch, that light fixture is going to go on. And then you turn on another switch, another light fixture will go on, and so on and so forth. So think about the brain, right? Because the brain, what it's receiving is the electrical signals that flow through the tracks and the processes of the neurons, right? A network of neurons. And everything starts in the environment, internal or external environment, when there's a change, and then a receptor is activated. So you can think about the receptor as a light switch. So the receptor is turned on, then the electrical current that is activated by the activation of the receptor is going to follow a network of neurons that is going to, they are going to conduct that electrical current to a specific area of the brain. And that's how we can tell, we can differentiate between sensations. It's where that electrical current is going to end up. So they're neural networks that conduct the electrical current to different areas of the brain. So let's see where these sensations are going to end up. So here you see the eye. So the eye is a receptor. You have the photoreceptors at the retina. And when the photons hit those photoreceptors, they're activated. If the change in electrical potential is strong enough, if it reaches that threshold, remember minus 55 millivolts, then you have initiation of an action potential. And it's going to be conducted first to the thalamus. So this area here in yellow is the thalamus. So all electrical current, all the electrical impulses that are generated by receptors in the outside or in the inside the environment are going to go through the thalamus, except for olfaction. So you see here the nose. So you have the olfactory receptors, and the action potential that is generated by the activation of the olfactory receptors doesn't go to the thalamus. It goes directly to the olfactory cortex, to the brain. So sometimes if one wants to deliver a drug directly to the brain, you can use a nasal spray because you know that this is going to go directly to the brain without going through the thalamus. So the eye, the action potential goes to the thalamus, and then from the thalamus, so remember, these are wirings. You think about a wire. It starts in the switch. The switch is the receptor, the photoreceptors. So the action potential, the electrical current is going to go to the thalamus. From the thalamus, it goes to the visual cortex at the occipital lobe. So the electrical impulse that ends up at the visual cortex at this area is perceived by the brain as coming from a visual stimulus. So the visual cortex is right here. So these are the primary sensory areas. That means the areas in the brain that receive the signals from specific receptors distributed throughout the body. So for example, taste, right? The taste receptors are located at the tongue. So when an action potential is generated, here you go to the thalamus, right? The wiring, the network of neurons goes to the thalamus, and then it goes to this small area here in the parietal lobe, and that's the gustatory cortex. Gustatory is for taste. Now let's think about sound. So the ear, inside you have the hair cells, which are the receptors for sound. So when they are activated, the action potential is going to go to the auditory cortex. This is located in the temporal lobe. Now, how about sensations that are activated by touch, body position, pain, temperature? These are somatic senses. Where are they going to end up? So they go into the thalamus, and then they go into this sliver, this slice of the parietal lobe that's known as the somatosensory area. So we talked about visual, auditory, gustatory, olfactory, and then these somatosensory areas. So these are the primary sensory areas of the brain. But you see that there's a lot of uh, surface or areas of the brain that are not the primary sensory areas. And those are known as association areas. And we will talk about those soon. Okay, so you see, how do we know this? How do we know that the action potential generated by photoreceptors generate an action potential and it's going to end up in the visual cortex? However, if it's a sound wave, the action potential generated is going to end up in the auditory cortex. So this is discovered based on functional MRIs, right? You see which area of the brain lights up when there's a specific stimulus. So you see here very clear the auditory cortex, where the action potentials generated by the receptors for sound, they're going to conduct action potential here, and here's a visual cortex. So depending on where the electrical currents are going to end up in the brain, that's what allows us to differentiate between sensations. So that's the concept of modality, that each sensory neuron carries an action potential of a specific type of sensation generated by a specific type of receptor. Okay, now let's focus here on the somatosensory area, right? So this somatosensory area receives action potentials that are generated by touch, body position, pain, and temperature. So there's a question here. Do messages ever get sent to the wrong part of the brain? Exactly, of course, unfortunately. And that's these unusual or abnormal cases also allow us to determine which area of the brain is involved in what specific function. And if it ends up in a different area of the brain, if the network is misleaded and brings the action potential to a area of the brain where it should not be, there is no specific, or the response is not going to be correct. Okay, so now we are concerned about this somatosensory area. So here is a sliver, a slice of the brain in the parietal lobe, where all the receptors that respond to touch, body position, pain, changes in temperature, all those sensations are going to end up here. But so now you ask the question, so how do we know if it's coming from a finger, if it's coming from the mouth, if it's coming from the hand? How do we know that? Remember, everything is converted into an electrical current. So how do we know? Well, this area, the somatosensory area, is divided into specific regions where the electrical stimulus from a specific area of the body is going to end up. So we call that, you see here, this is the sensory cortex. So each area of the sensory cortex is going to receive an input for a specific area. For example, so this area here of the somatosensory area is going to receive input from receptors located in the mouth. And you see that the proportion that is dedicated to each specific area of the body does not correspond to the normal size of our body. Why? Because it depends on how many receptors. So we have lots of receptors in the mouth and in the face. We have many receptors in the hand. The trunk, there's very few receptors. So the area of the somatosensory area that corresponds, that's going to receive signals from the trunk is much smaller compared to the mouth because the density, the receptor density in the mouth is much greater. So any electrical signal that is generated by receptors that are located in the mouth, like, are going to end up in this area of the somato, in this specific region of the somatosensory area. So this representation of our body on the somatosensory area is known as the homunculus. It's an abnormal kind of individual, if you want. Now notice that the somatosensory area that is in the parietal lobe is right next to the motor area. Here, we haven't spoken about the motor area. We just now talking about the sensory cortex. But the motor cortex is right next to the sensory cortex because the action potentials are going to travel then from the sensory cortex to the motor cortex. So again, here you see the somatosensory cortex, and you see that each area, each specific region of this somatosensory cortex is dedicated to a particular area of our body. So look here, upper lips, the face, the eyes, the nose, the back. The back does not have that many receptors. So I hope you understand this. So within the somatosensory cortex, each part of the body is represented, and it's going to receive the action potentials that are generated by receptors in specific areas of the brain. And this is known as the sensory homunculus, or the little man. So the size of the sensory cortex that corresponds to a specific area of the brain, the size of it is corresponds to receptor density. So, for example, fingertips have many receptors, touch receptors. They contain about 100 times more receptors per square centimeter, 100 times, than the skin on our back. So, of course, we're much more sensitive with our fingertips than if something touches our back. So this is what is known as the sensory homunculus. Now, so we discussed the primary sensory areas that receive the action potentials coming from the periphery, and the areas that receive the action potentials, where these action potentials are going to end up. How about now the motor areas? Well, the motor areas bring the information from the motor cortex down to the spinal cord, and then that's going to be conveyed to the periphery, to the effectors. Remember, what are the effectors in this case? They're the skeletal smooth and cardiac muscle and the glands. So the motor areas, there are only two, the primary motor area and the Broca area. So why is it called Broca? It was Mr. Broca, or Dr. Broca, that discovered that this area here, located in the frontal cortex on the left side, this is not symmetrical. We only have it on the left, usually on the left hemisphere, is dedicated to establishing which muscles are going to be used for speech. So it's a motor area because it controls, it makes the plan of which muscles are going to be used to say a particular word or to read a particular sentence. So that plan that allows us the language, the speaking language, is produced here in this area known as the Broca area. All the other movements are located or here in the primary motor area. So you see that from the sensory cortex, the action potentials travel to the motor cortex, and then the action potential is going to travel down, for example, the pyramidal tracts or the extrapyramidal tracts, and then they're going to go down to the spinal cord, and from the spinal cord down to the out to the periphery. Okay, so this Broca area is very important for language, but it's only involved in the performance of language, in the speaking language. To understand language, there's another area that is involved, and it's known as the Warnick area. And we will discuss this when we talk about the association areas. So what am I showing here? The visual cortex, primary visual cortex, the auditory cortex. So you know that both the visual cortex and the auditory cortex can lead to language, to a response in language. So there has to be a connection between the auditory cortex and the visual cortex and this area, the Broca area and the Warnick area. So these areas, Broca, Warnick, auditory and visual cortex, they're all interrelated in the process of language, understanding and speaking language. Both the Warnick area and the Broca area are most often located in the left hemisphere. So here you have also the motor homunculus. So it corresponds, the area that corresponds to the muscles that are controlled depends on the area, the number of muscles in the specific area of the body. So there is a sensory homunculus and a motor homunculus. And you can see that the sensory cortex is very close. The sensory cortex located in the parietal lobe, the motor cortex located in the frontal cortex. So from here, the action potentials travel down the tracts, the pyramidal or extrapyramidal tracts down the spinal cord. Okay, now we move on to the association areas. So the association areas are not dedicated specifically to receiving information, action potentials from receptors. What they do are to transmitting to specific effectors. So they are in between. They're in between the sensory and the motor areas. They are dedicated to interpretation, to cognition, to memory. So those are known as the association areas. So they associate the sensory information with the motor response. So in the association areas is where interpretation occurs. And what is interesting is that the size of the association area increases with increased cognition, with increased intelligence. So you see here the brain size compared rat, cat, chimpanzee, and human. And look at the motor areas and sensory areas. They change in size, but not that much, right? So you go from the motor areas are purple. You see the rat, the cat, chimpanzee, and human. And then you see the sensory areas represented in green, rat, cat, chimpanzee, and human. But now look at the association areas, the areas where there is cognition, where there's memory, where there's creativity. And look at the size. The rat is minimal. The cat, you have a little bigger. The chimpanzee, much bigger. But now the human is bigger, is the biggest of all. So that's why the cognition, imagination, intellect is so much more developed in the human brain than in the other species. So it depends on this association area that makes the connection between sensory information and motor response. So the association areas, they are dedicated to specific sensation areas. So they're close to. So we have the somatosensory association area. What does it do? It interprets and stores memories. The visual association area. So this visual is not the primary visual area that receives action potentials directly from the photoreceptors. This is the visual association area. That what does it do? It relates past to present. So it remembers memory. Auditory association area is involved in speech. We talked about the Warnick area, noise and music. The gnostic area, that's also known as the Warnick. So both Broca and Warnick, those names come from the investigators that discovered that these specific areas were dedicated to that function. So the Warnick area integrates and interprets the language. Premotor association is involved in learned skilled movements. Remember how to ride a bicycle, how to play tennis, how to play soccer, how to play the violin. And then this area, which is very interesting, the frontal eye field that allows you when you are reading something, let's say you're looking for a specific name on the list of 100 names. And you can just skim down the list very quickly with your eyes and until you reach that specific name. So that is known as the eye field, frontal eye field, gives you the ability to scan documents. It's a scanning eye movement. So it's different than the response to a visual clue. So this is interpretation, right? You're allowed to, from going very, very quickly down the list of 100 names, to pick the one that you are interested in. So this is what is shown here, the association areas. So this is the somatosensory primary area and the primary motor cortex. But you see that there's a lot of area in the brain that is dedicated to the association, to the interpretation of the signals that come from the periphery. So I was talking about the frontal eye field. So how is this network? What is the pathway that of the action potentials that are generated at the eye, at the photoreceptors? So you know that they should go directly to the visual cortex, right? So here are the photoreceptors are activated. Then you have the optic nerve, the action potential that is conducted to the primary visual cortex. It's at the occipital lobe. But for this property, the ability to scan, then it goes to the posterior parietal cortex. This is involved in attention, so association area, right? Then it goes to the frontal eye field, here in the frontal cortex. And then it goes down. So a different pathway from just simple vision, the ability to interpret, to go quickly down a list. So it's a very important and intellectual function. So the networks, the pathways are different. So here again, you see the different areas of the brain. Is the Warnick area. involved in language interpretation in the temporal lobe. And here's the Broca area that decides what muscles are going to be used to read aloud a specific text. So there has to be communication between these two, the interpretation of language and the generation of language, of speaking. So this slide really tells you in detail what are the functions of the different areas of the brain. And you see here, this is the primitive brain, the brain stem, that controls the heart rate, respiratory rate, and so on. And here's the cerebellum. Cerebellum can be considered an association area, and it's dedicated to coordination of movement. It contains a lot of Purkinje cells. Remember, those that can have more than 200,000 synapses. And these Purkinje cells, they are GABAergic neurons. They have the GABA neurotransmitter, which is inhibitory. So it's highly enriched in neurons that contain the GABA neurotransmitter. Okay, now one can take advantage of the electrical signals that are generated in these different areas of the brain to overcome deficiencies. So, for example, in this case, let's say that this individual lost the two arms and it had a prosthesis. So you have two mechanical arms that were attached to the body of this individual. And they are controlled by the brain activity. So how is this possible? How is it possible that the brain can control the movement of these mechanical arms? So the neurons are rerouted. So instead of going to the previous arms that were here, they are rerouted to a different area, to the chest, and from there, so the motor cortex, the action potential goes to the shoulder. From the shoulder, they're rerouted to electrodes located in the chest. And then those electrodes conduct the action potential to the artificial arms. So this is really amazing. How can we have the messages that are initiated in the brain being captured by electrodes and that will, through a different wiring, control the movement of a mechanical arm? So it's taking advantage of the electrical signals that are generated in the brain to move a mechanical arm or a mechanical leg. So take advantage, you reroute those action potentials, you take advantage of the neurons that are still intact to conduct those action potentials, in this case to the chest, where there are electrodes that are activated, and that electrical signal now is conducted to the mechanical arms. The same here for individuals that are blind. So there's a camera that is implanted like on glass, and it's connected by electrical signals to the visual cortex. So this is still in very, very initial stages, but it allows some blind people not to hit a table or hit a door or something like that. So it gives some partial visual signals. So the action potential is conducted via electrodes and artificial wirings directly to the visual cortex. So it's from the eyes to camera and to the visual cortex. Now here I'm going to show you how a person can make a wheelchair move not just by um, initiating action potentials in the brain. So this is a cap that has electrodes, and it's placed over the brain of an individual. Now, what you're going to see is this individual is controlling the movement of the wheelchair based on the voluntary action of the brain, of different areas of the brain. Remember, it's the motor cortex. So by activating the different areas of the brain, then you have electrodes that will conduct that electrical impulse to a computer, and the computer will make the chair move in the appropriate direction. So what's happening here is you have the interplay between the electrical signals that are generated in our brain and that are connected to an electrical current conducted by wiring. It's not neurons, artificial wiring, and that electrical signal is going to allow either for mechanical arms or for vision or for movement of a chair, depending on which area of the brain is going to be activated. So, of course, this is becoming more and more evolved, and who knows, it's a lot of promising future applications. So he's controlling it with his mind. Exactly. How does thinking lead to the generation of an action potential? Exactly. Thinking is an electrical signal. It's specific neurons that are activated. And what do we mean by activating a neuron? Activating a neuron is the generation of an electrical signal that can be picked up by the electrodes. So that's the beauty. So the electrical signal in the neurons is generated by ions. In the wiring, the artificial wiring, the electrical signal is in electrons, but they are connected. The electrical signal generated by the neuron is going to be connected to the electrical signal. It's going to travel through the wires. So it depends on the electrical signals that are generated by the brain. It's amazing. Okay, now let's talk about language. How is this possible? How is it that an electrical signal can lead us to properly read the text, or for you to hear me, and if you wanted to repeat what I said? What is the network? What are the pathways that are involved in these actions? Okay, so you know that there are these two specific areas of the brain that are asymmetrical. They're only located either on the left hemisphere or on the right hemisphere. Most cases, it's on the left hemisphere. So you have the Broca area that is in the frontal lobe, and this is where the neurons can generate a signal that's going to control the proper muscles to generate the speaking language. And the Warneck area is the neurons are going to give us the ability to interpret the language, know what it means. And knowing what it means, then the electrical signal is going to be conducted by this arcuate fasciculus. So these are the white matter, the myelinated axons that connect the Warneck area with the Broca area. So the electrical signal is going to be conducted from the Warneck area all the way to the Broca area. And then from the Broca area, it's going to go to the motor cortex. And from the motor cortex, it's going to come down to the spinal cord. And the signal is going to end up controlling which muscles, which skeletal muscles specifically, are going to be used to say a certain word and so on. So the Warnick area allows us to understand language, and the Broca area allows us to speak language. So one student asked, so what happens if one of these is not working properly? So this leads to a condition known as aphasia. But let's see what is the pathway that is involved. So let's say you want to read a text out loud, right? So you're looking at the page of a book, and the specific photoreceptors are going to be activated. The action potential is going to be conducted to the visual cortex. So now we have an electrical signal generated by the neurons and conducted by the axons, right? It's going to go directly to the visual cortex. So this is sensory. From the visual cortex, it's going to go to the Warnick area. From the Warnick area, it's going to go to the Broca area. And from the Broca area, it's going to go to the motor cortex. From the motor cortex, it's going to come down the spinal cord, and the signal is going to end up in specific muscles that will allow to read out loud whatever is on that page. So this is the pathway that is going to be followed by the action potential. So speaking a written word. So speaking a written word, why is written word? Because you're using the photoreceptors, you're using the eye. What happens if you want to repeat what you are hearing? So the pathway is different. Here you have the photoreceptors conducting the action potential to the visual cortex. Now, how about hearing? Now you have the hair cells, the receptors for auditory stimuli. The action potential is going to go to the auditory cortex in the temporal lobe. From the auditory cortex is going to go to the Warnick area. So here the difference is the visual word, the visual sensation goes from the visual cortex to the Warnick area. The auditory action potential is going from the auditory cortex to the Warnick area. Then it's the same. From the Warnick area goes to Broca, from Broca to motor cortex. So when learning a new language, is it easier to understand the new language first, and then comes the ability to speak it? Is it because the person needs to create new pathways from Warnick to Broca area? So to learn a language, a new language, the area of the brain is going to be different because this is new. And then when it becomes established, becomes part of the normal language, then it moves to another area of the brain. And what is interesting is, like, I'm originally from Portugal, and I've been living in this country for 50 years, but I still have an accent, right? I'm not very musical. You know, I used to put my children to sleep. I would try to sing to them, and they would say, please stop singing, because I'm not very musical. But I'm here for 50 years. I know I speak Portuguese at home, but I could have lost my accent, but I didn't. Why? Because they say that when you first speak a language, those neurons, because remember, neurons are permanent. There's no neurogenesis as you age. So those neurons establish an accent that's very hard then to change. If you hear the two languages at the same time, then you don't have that problem because the neurons that are being activated start like this. But I started with Portuguese, and then much later I had English. And it's really amazing because if you think people that speak different languages, like I can be speaking with my sister in Portuguese. And if someone walks in, I can speak immediately in English. I think that this is amazing that it's so, because it's two different areas of the brain that are involved. But I'm not a language expert. So, question here, just to be sure, the action potentials go to the thalamus first, from the hair cells, yes, and photoreceptors, yes, absolutely. You're absolutely correct. Everything except for olfaction goes first to the thalamus. But I just shortened that that circuit, that pathway. So we were here. Let's see. So why doesn't neuroplasticity allow us to lose our accents? What makes accents so hard to lose? Because they become, remember how the synapses change? You have synapses that are short term, and there are synapses that are long term. So if those synapses now become long term, then that plasticity is lost because those synapses now become permanent. They're very strong. So that's why. You can learn the language, so these areas can adapt, and the different area of the brain will be dedicated to a different language. But the accent is strong, very strong. The synapses are very strong. So it's the concept of long-term memory, of synapses that physically change in size. And the microglia, remember the microglia are the cells that destroy synapses that are not strong enough. So when we sleep, the microglia are very active, removing synapses that are weak, because we don't want to keep all the synapses that we establish during the day. So if the synapse isn't strong, the microglia cut it, making it harder to learn a second language. Well, how do you learn a second language? Repetition, repetition, repetition. So now the synapse now becomes very strong. If it's weak, then you won't remember, right? And as you grow older, the synaptic plasticity also decreases. Remember, when you're a child at around 6 and 7 years old, the brain is highly plastic. It can learn languages very quickly because the synapses are not yet formed, many of them. So you have high levels of brain plasticity. If you grow older, then it becomes more difficult. Okay. Now let's see which areas of the brain. Remember, I talked to you about the functional MRIs that show you areas of the brain that light up when you're doing a specific action. So when you're hearing, you can see that the auditory cortex and warning, they are both located in the temporal lobe. They light up. If you are seeing, if you're involved in activity that involves seeing, the photoreceptors, see what lights up the occipital cortex. When you're speaking, you have the Broca area and the motor cortex. So you can see very specifically when you're doing a certain activity that there are areas of the brain that light up that correspond to where those action potentials that are generated by the receptors, where they're going to be conducted, where they are going to end up. If one area is stronger than the other, will it light up more versus other areas? Yes, that's the whole idea. So here I should have explained that the red signal represents the highest activity, the highest electrical potential. So you see that these areas are where the activity, the brain activity is the highest. So speaking, seeing, the occipital lobe, the temporal lobe for hearing, and Broca and motor cortex. Like right now, I'm talking. So what do I have? I have the Broca area and the motor area are highly active. Okay, so what happens if the Broca area or the Warnock area are damaged? Then this leads to language problems that are known as aphasia. So aphasia is impaired language. And there are two different types of aphasia, the Broca aphasia and the Warnick aphasia. So you see here the Warnick area and the Broca area. So the language goes from the visual cortex to the Warnick area, Broca area, and then motor cortex. So if the Broca area is affected, then you can still understand language, but you won't be able to say it perfectly. It comes out with no meaning, no sense. It's like a word salad. If the Warnick area is damaged, then you still will be able to say the words properly, but they don't have any meaning, the correct meaning. So if there's an aphasia related to damage to the Broca area, you can still understand the language. The person can understand the language, but the speech is slow and slurred. Why? Because there is no perfect plan to come out with the perfect words. The muscles, the necessary muscles, are not controlled properly. If the Warnick area is affected, then the person can speak clearly because the Broca area is still intact. But the words have no sense. They make no sense because you don't know what you're saying. You're saying it perfectly, but you don't know what you're saying because the Warnick area is damaged. Okay, so let's move on to brain lateralization. So we have two hemispheres, and they are not the same. They're not symmetrical. For one, you already know that the language centers in most people are located in the left hemisphere. But for each person, one of the hemispheres can be dominant. So the right hemisphere, the different areas, association areas, remember, they are mostly involved in artistic functions. So art awareness, imagination, music. And what is interesting is if the right hemisphere is the dominant hemisphere, these individuals many times are artists and they are left-handed. Why? Because the right hemisphere controls the left side of the body. Remember, there's the decussation of the pyramids. There's the crossover. If the left hemisphere is a dominant hemisphere, then these types of functions are located in the left hemisphere. The person is right-handed, but the scientific thought, spoken language, reasoning, and so on, those areas, those functions are located in the left brain. And these individuals are right-handed. So, of course, this is very easy to explain, and it's not 100%. It's not like every person that is has the right hemisphere is the dominant is going to be left-handed. It's going to be an artist. This, of course, there's a lot of mixtures, and there are individuals that are ambidextrous. That means that the two hemispheres, there's no dominant hemisphere. Okay, let's see. So, does the duck mean anything here? Yes. So if the Warnick area is affected, you see the duck, but you don't understand that it's a duck. So you want to say what it is, but you can say the word duck, but it's not, it doesn't mean, it doesn't have the right meaning. And this is what I usually also bring into the discussion is how can you have one word, the same word, duck, being a verb or a noun, right? To duck, remember to hide is a verb, but the duck is a noun. So this grammatical knowledge is located, is possible because of the Warnick area. So let's see here. What type of treatment is done for aphasia? Well, you have a lot of language therapy that can be used. There are people that are language and help individuals that have language problems. Are more people left brain since most people are right-handed? Yes, that is a fact. That's a funny question. Will the dominant side be more wrinkly brained? Well, the folds and sulci, they're, there's so many different functions and that are involved. I don't think that can be. So because I'm saying it's the dominant side, so it could be larger. I don't think it has to do with the size of the hemisphere, more with the quality of the functions. But it's a good question. Okay, so we know that there's crossover information. And why does this happen? Why is it that our left brain controls the right side, and why does the right brain control the left side? What is the advantage? Well, this is still under study, and it's very hard to research. And it seems that throughout evolution, there was this 180-degree switch that happened. And there's all kinds of hypotheses that are involved explaining why this happens, why there's this crossover. Can you develop artistic abilities by using your left hand, for example? Well, you can develop the accuracy of your left hand. Let's say you're right-handed and you damage or you lose your right hand in an accident. Then you can train your left hand to become more accurate. But switching the overall dominance of the hemisphere is hard. Okay, so we have this crossover, and what the slide here represents is the eye with the photoreceptors on the retina, and you see that the optical nerve conducts the action potential to the opposite side. And here you see the same. And then the motor action potential travels also. Remember the decussation of the pyramids. So the optical nerve allows the sensory, the action potential activated by the photoreceptors through the sensory neuron. The optical nerve is sensory, it's conducted to the opposite side of the brain, but then the motor action potential also goes to the opposite side because of the decussation of the pyramids. Remember the motor tracks that will cross over at the medulla. So we have the right side, the right brain controlling the left side, and the left brain controlling the right side. And how do we know this? Here you see the corpus callosum, right? So these are the nerve fibers that allow for communication between the two hemispheres. So these are myelinated fibers that conduct the action potentials from one hemisphere to the other. So what happens here is sometimes epileptic patients, the corpus callosum is severed. And some kind of experiments can be done. For example, you have an individual where the corpus callosum is severed. The optic nerves are still intact. So if the corpus callosum is severed, that means that there's no communication between the right and left side of the brain, correct? Only through the optic nerves. So now you have a screen and you put a fork on the right side, and you ask this individual, you cover the left side of the left eye. You ask the individual to look at the screen, and you ask the individual to tell you what do they see. So spoken, right? The spoken language. So the action potential comes through the right eye. It travels to the left hemisphere. It can travel to the left hemisphere because it goes through the optic nerve, sensory information, goes to the Broca area, the language center, goes to the visual cortex, and then to the language center, which is located on the left hemisphere. So the individual very easily will tell you it's a fork. So remember, it's the stimulus comes through the right eye. It ends up on the left hemisphere, so the individual can perfectly tell you that it's a fork because there's no need to communication between the right and the left hemisphere. However, now let's say you cover the individual's right eye and you have the fork on the left side of the screen. And now you ask the individual not to tell you, not to speak what he sees, but to point to what he sees. So you show the individual a knife, a fork, and a spoon, and you ask the individual to point, no language, movement. So what happens here? So you have the action potential coming through the left eye, goes to the right hemisphere. So that in the right hemisphere, there is no language center. So the individual could not tell you that it's a fork. But now you're asking the individual to point. So it can point because you have here the motor cortex. So you don't need information coming from the left hemisphere. There's a motor cortex is present on both hemispheres. So the individual can point to the fork, but it cannot tell you. There's no verbal response because there is no information going from the right to the left. So the language is not going to be activated. But the motor cortex can be activated because you have the action potential going to the occipital lobe, and then to the motor cortex. So the individual perfectly can tell you that it can show you that it is a fork, but it cannot tell you that it's a fork. So in general, the person can speak, but just not know because their right eye is closed. Right, exactly. Exactly. That's a must. The right eye is closed. So you don't have any information going to the language center. Exactly. And there's also these figures that are known as chimeric figures. So you have half of a figure is a woman and half of a figure is a man. And either you ask the individual to tell you what is the individual seeing, or to point to what the individual is seeing. And if you ask the individual to tell you to speak, the individual will tell you that it is a man, right? Because this information comes from the right eye. Now you have the two eyes open, and this information comes through the right eye to the language center. So the individual is able to tell you that it sees a man. But if you ask the individual to point to what it sees, the information comes from the right eye and goes to the occipital lobe and then to the cortex. So it points to a female. So this leads to a condition known as the disconnection syndrome, when there's a problem in the connection between right and left hemisphere. And there's another condition known as neglect syndrome. And these individuals have a problem in the right parietal lobe, so that they neglect, they forget that there's something on the left side. So you show a picture to the individual and ask this individual to copy this picture. So the individual is only going to draw what is on the right side, because there's no information coming to the left parietal lobe. So these are lesions that are localized in the right parietal lobe, that what they do is that the individual neglects, forgets, doesn't see, doesn't react what is on the left side. So, for example, a person that suffers from neglect syndrome might only put makeup on the right side of the face and neglect the left side. So it's kind of a condition where the one side of the hemisphere in the parietal lobe is damaged. And usually this occurs in the right parietal lobe. Okay, so we covered the cerebrum, all its functions, and we still have a lot to learn, and to especially to be able to overcome so many deficiencies that people suffer from as we learn more and more and take advantage of these electrical signals, and to be able to have chips put in the brain, and all kinds of strategies. Okay, now let's move on to the diencephalon. So we forget about the cerebrum, we leave that, and now we're going down to the diencephalon. The diencephalon is made up of three regions: the thalamus, we talked about the thalamus a lot, the hypothalamus, and the pineal gland. So these are the three components of the diencephalon. So let's start with the pineal gland. So it's a small gland located here near the, so we're talking now going deep into the brain. And the pineal gland is a gland that produces melatonin. And when there's light, the pineal gland is inhibited. So there's no melatonin being produced. And melatonin is associated with promoting sleep. So the sleepiness state. So if there's light, the pineal gland is inhibited. In the dark, the pineal gland is active. So what happens here? So you have, let's say there's light, the photoreceptors are activated. Here you see the optic nerve brings the action potential to this area in the hypothalamus that's known as the suprachiasmatic nucleus. Suprachiasmatic nucleus receives the information, the action potentials from the photoreceptor. Then the action potential continues to this this area, that's the superior cervical ganglia. So this is part of the sympathetic nervous system. Remember, ganglia, superior cervical ganglia is part of the sympathetic nervous system, where norepinephrine is released. So norepinephrine, then you see the sympathetic neuron going to innervate, conducting the action potential. Remember the preganglionic and postganglionic. So we're talking about the sympathetic nervous system, where the preganglionic neuron is short, the postganglionic neuron is long. So you have the action potential coming from the hypothalamus, going to the superior cervical ganglia, and it continues to the pineal gland, where norepinephrine is released. And norepinephrine now inhibits the pineal gland. So there's no melatonin being produced. So the action of the photoreceptors at the retina initiates an action potential that is going to inhibit the pineal gland. If there is no light, then the pineal gland is active, and then melatonin is going to be released. Okay, now let's concentrate on the thalamus. So we talked a lot about the thalamus. So to have an active thalamus is essential for us to perceive sensations, to perceive changes in the external and internal environment. But it's not essential for life. As long as we have the brain stem active, that means that our respiration, our heartbeat is still intact. We can still be alive, but we will not respond to changes in the environment. So we live in what is called a vegetative state, because all the action potentials that, except olfaction, that are generated in the periphery, they're going to end up in the thalamus. So if the thalamus is damaged, then we won't respond. We won't be aware of any changes in the environment, but we can still be alive because our brain stem is still intact. And how is this possible? So how did we, some examples of individuals that are living in a vegetative state. So this young woman, she was 24 years old, and she took a mixture in a party, a wild party. She took a mixture of drugs and alcohol, and specifically her thalamus was damaged, stopped working. So this person, her name is Karen Ann Quinlan. She was in a coma from 1975 to 1985 for 10 years. So she was first connected to a respirator, and then the family decided, because she didn't respond to any stimulus. You could touch her hands, touch her face, tickle her feet. She wouldn't respond. She did not respond to any sensation, but she could live. Why? Because her hindbrain was still intact. So she could breathe, her heart was beating, and so on. And the family, after a few years, they decided that they wanted to remove the respiratory machine. But Carrie Ann continued to live for 10 years without any respiratory machine. She lived in a coma, in a vegetative state, because her thalamus was gone. The same happened to this other woman, Terry Schiavo. She had a stroke that destroyed or induced a malfunction of the thalamus. So again, she lived. She didn't respond to any sensation, but she was alive for 13 years. So this shows that we can live without, if you call that living in a vegetative state without the thalamus. Okay, let's see what time is it. We go a little more for I want to discuss the hypothalamus. So we talked about pineal gland, the thalamus, and the hypothalamus. So the hypothalamus is a much smaller area than the thalamus, but it's very important. It has many nuclei. Remember what nuclei are in the brain. These are gray matter areas within the white matter. So the hypothalamus is a very important area, and it regulates many functions. It has all these nuclei that are here represented in different colors, and they play roles in long-term memory. The arcuate nucleus secretes hormones that then are released into that control the anterior pituitary. The supraoptic nucleus releases antidiuretic hormone. Paraventricular nucleus secretes oxytocin. The preoptic nucleus, remember we talked about this way in the beginning of the semester, that this is the thermostat of the body, and this relates to temperature. It also regulates the autonomic nervous system. And now we add one more nucleus, the suprachiasmatic nucleus that sends signals to the pineal gland. So it's a very important area, and it's the intermediary between the nervous system and the endocrine system. So it's part of the brain, but it also releases hormones that control the pituitary gland and other areas. It's part of the limbic system, so it's involved in emotion. It controls body temperature, food intake first, and waking and sleep patterns because its relationship to the pineal gland. So you see that the hypothalamus is very, very important. It's a small area, but it contains these nuclei that are centers for controlling many different functions in our body. And one, for example, is the feeling of thirst. So it is dependent on the hypothalamus. So there are osmoreceptors located at the hypothalamus, and they shrink when there's lots of water. This activates the hypothalamic osmoreceptor neurons. They secrete ADH, which is the antidiuretic hormone. This hormone then goes from the hypothalamus all the way to the kidney. It's also known vasopressin or antidiuretic hormone. And this prevents water from being excreted into the urine. So you have lots of water resulting in the release of a hormone from the hypothalamus that goes all the way to the kidney to prevent loss of water, to prevent dehydration, more dehydration. Okay, now the last area, we'll go just a few more minutes, the cerebellum. This is the area of the brain, we call it the little brain, that is tucked down in this area near the hindbrain. And it has the left hemisphere, right hemisphere, and the vermis in the middle. All of these contain gray matter and white matter. And in the cerebellum, the gray matter is on the outside. So the left hemisphere, right hemisphere, and vermis, they all have gray matter on the outside, just like in the cerebrum, and the white matter in the inside. The white matter contains those Purkinje cells that are highly branched and can contain up to 200,000 synapses. And they are very elaborate. And um, so this is what said here. Here are the Purkinje cells. And the Purkinje cells, the neurotransmitter is GABA, is GABAergic neuron. So you need, this is what the break that is necessary for controlling, for highly controlled and specific muscle movement. So if the cerebellum is affected, like by alcohol, you lose balance. So this is known as ataxia. So ataxia is related to malfunction of the cerebellum. Okay, and I think that's it. So there's a question here. Why do people say that coma patients can hear what is said to them by friends and family visiting? Well, you never know, right? Because they cannot respond. So if the thalamus is damaged, then the the action potentials will not travel to the whole brain. But you don't know if it's the whole thalamus that is going to be damaged. Maybe there's some partial area of the thalamus that can still conduct the action potential to the auditory cortex. So you never know because people cannot react. That's it for today. I hope you had a good time. I did. And I will see you on Thursday.
Professor, I have a question.
Yes.
I emailed you earlier today regarding about my exam two. I wasn't able to see, well, I'm still not able to see it on Brightspace, my score.
Oh, you sent me an email?
Yes.
When?
I believe around 3 o'clock today.
Um, Let me see. Can you tell me your name, please?
Nirvana.
Oh, yes, I haven't read it yet. I will reply.
Okay, perfect. Thank you.
I see your email. You know, I get lots and lots of emails, as you can understand, but
Th-
I will reply to it for sure, okay?
Don't worry, I understand. Thank you.
Okay. Okay, bye-bye, everybody. See you Thursday.